Hello again. Well, I've just realized that I forgot to show you the diagram in the previous video about quorum sensing. And the diagram, as seen from above downward, shows here that the the bacterial cells starts producing autoinducer molecules. The language used by the bacteria to communicate is through chemical molecules. And then these molecules, intracellular autoinducers, will diffuse to the outside or will be actively transported to the outside. And the cells continue to divide and meanwhile continue to produce autoinducers. As you can see here, the extracellular concentration of the autoinducer is smaller than the intracellular concentration. Now, when the cells are replicated and they continue forming autoinducers, there comes a time when the concentration of the autoinducer outside becomes equal to the concentration of the autoinducer inside. And then they continue formation of or synthesis of the autoinducer until the extracellular concentration outside becomes larger than the intracellular concentration. At that time, the cell has a solution. It can actively transport the autoinducer to the outside, but this would be too energy costly for the cell, and therefore the autoinducers are no more diffused or actively transported to the outside, and they start accumulating inside the bacterial cell, and the higher autoinducer concentration inside will auto-regulate itself and will down-regulate itself by binding with the receptor, inhibiting the transcription factors that transcribe their own genes that synthesize them, and therefore inhibit their transcription and translation. This is down regulation. If they want to limit a process like limiting mitosis or ending bioluminescent pigment production, or ending division to form a biofilm. But if they are still inside the process of making a biofilm or making bioluminescence, then the accumulated autoinducers inside would not down-regulate, but on the other hand, would up-regulate and increase their concentration, increase gene expression, the genes for synthesis of the bioluminescent pigment or for growth factor and mitosis, as we have explained in the previous video. And that's it. This was a very short extension for the first video on quorum sensing, and see you on another one topic video very soon. Thank you.